Hi, I'm Richard Sever from Open Archive and Cold Spring Harbor Lab. With me I have Coleen Murphy from Princeton. We're here at the annual Symposium at Cold Spring Harbor Lab. This year it's on senescence and aging. Coleen, welcome to Cold Spring Harbor.
Thank you.
So you gave an amazing talk the other day about aging and specifically memory loss in nematodes. And what I thought was kind of nice at the beginning was you preempted one of the criticisms, which was, okay, we want to know about aging and memory in humans. Why nematodes? So can you sort of just justify the nematode for me?
Sure. So beyond being just a very excellent model for aging, of course, we could say, well, worms don't have, normal, they're not solving calculus problems or things like that, right? But every organism needs to figure out how to survive in their environment. And as our lab has shown, they actually use very similar mechanisms to learn about their environment, so Pavlovian associations, and to learn, here's where food is because I can smell it, and I get rewarded by going to that smell by eating food, and so we could use that to turn it into a memory assay. And of course, the reason that's important is because evolution happened, so all those things that we care about in neurons, most of those proteins have the same proteins in us, and so you can use the nematode to actually study a very seems like it would be really complicated to do in humans, very slow, see what happens with age. You can do it really quickly in a nematode, and you can actually end up finding the same mechanisms that are used in mammals to carry out learning and memory and that decline with age.
Right. So I mean, so it's interesting. You mentioned Pavlov's dogs. So what's we all know the Pavlov's dog assay. What's the assay that you use in nematodes?
All right. So since they are pretty simple, we basically leverage the thing that we know that they care about. They care about food. In this case, bacteria. Finding bacteria to eat, and their senses are basically olfactory. So they smell their world, and so by pairing those two things, the smell of a chemical that they kind of don't care about when at this particular concentration, and pairing that with food, we can then ask, did they learn this association? So this Pavlovian association between food and the odor.
So it's a smell they never normally encounter in nature.
Well, I think most of what we've figured out that they react to in the lab are actually things that they might smell. They might be bacterial metabolites.
Oh, okay.
So that's why we think that they have any sort of ability to detect them.
Right.
And so other people, we actually based our work on earlier work that others had done to figure out, if you screen a bunch of chemicals, which things do the animals react to? Right. So at higher concentrations of butanone, they actually are more attracted to it. So we knew that they use a particular neuron to sense that chemical. So that was work that was done by Corey Bargman's lab many years ago. And so just saying, okay, well, those are the building blocks. We can develop an assay that might better mimic the kind of learning that we care about in humans.
Right. So butanone was the chemical that
butanone was the chemical in this case. You can do it with a lot of different chemicals. We just used that because we could actually tune the concentration to be something that was neutral. Because you kind of need to start with something neutral, so then they can, under untrained circumstances, they just kind of are, they're apathetic about it, and then make them care more about it by pairing it with food.
Right. Oh, I see. I see.
I was going to say there's other chemicals that they are strongly attracted to, and you can do more of a negative type, like a PTSD type of learning and memory, by pairing that with starvation.
Right.
And so when I started the lab, what I really wanted to do is focus on the kind of learning and memory that we care about preserving with with age, which is different from that you try to want to get rid of, like the PTSD. So then there's some overlap between those mechanisms, but we really wanted to focus on this particular area that we knew declines with age.
Right. So that's the key, is where you get that you mimic the sort of decline in memory with age.
Yeah. And it also turned out to be a really good. This is when I started my lab in 2005. My very first graduate student working on that project, Mandy Calfin, she and I designed this particular assay, and it was kind of a gamble at the time because nobody thought that the worms could actually have any kind of long-term memory. But it turns out that's really worked out well, and the mechanisms are so well conserved that we can learn things that actually apply to higher organisms.
Yeah. Well, I mean, that was what was interesting to me, not being a nematode expert at all, but suddenly hearing you talk about Krebs transcription factors, histone methylation, and various other pathways. So as you started picking apart the sort of molecular signaling involved, what did you find?
Right. So it's a little bit of chicken and the egg, because in order to call it actually learning, memory, like short-term memory, long-term memory, we knew that we had to be able to check the boxes. So once we started developing the assay, we were really relieved to find out that of the things that were already known to be involved for like learning, memory, long-term memory in other organisms like flies, aplysia, mice, those were all involved and required for C. elegans memory. So that already like gave us a platform to say, okay, we know this is the same kind of thing.
Was that like synaptic proteins, that type of thing?
A lot of signaling molecules, and CREB, of course, being the transcription factor, is really important for long-term memory.
Right.
So the surprise for us has been that actually CREB is not just important for the activated transcription that's happening during long-term memory formation, but the animals use CREB, the transcription factor CREB, to turn on a bunch of synaptic genes, things that are functioning in the neurons, to also allow them to have normal short-term memory.
Right.
And so that was kind of cool to discover. And actually I had to think about, like, change the way I thought about that transcription.
So that was unexpected, the short-term
Yeah, in fact, actually, that was, and for some reason, for years, we knew DAF2 worms had better short-term memory. And because we had already...
These are the aging mutants?
The DAF2 insulin mutants actually double their lifespan, and they have longer than that, like a longer memory span than their lifespan.
Wow.
Yeah. And so we think of them as extremely healthy, and so a really good model to figure out, like, well, what do they do better that we could then co-opt, right? And so this idea that CREB would be also required for DAF2's ability to carry out memory longer than wild-type worms was really kind of mind-blowing, because we kind of shoehorned CREB into this like only for long-term memory kind of box.
Right.
And so this said that actually it's important for all these things, and it becomes even more important as the animals age. They kind of transition over to an entirely CREB-dependent, like CREB is important for those long-lived DAF2 animals to carry out memory with age, even more important than it was when they were young.
Right.
So it kind of does this transition. So I had to kind of change the way I thought about what DAF2 animals were doing as a result of these experiments we did in the lab.
Yeah, well, it's nice when that happens. It's nice when that happens.
Although I think we would have done the experiments earlier if we had been thinking that way.
Right. Yeah.
It's kind of crazy that we knew that DAF2 worms had a really long memory for since 2008. But we never bothered to ask, is that CREB dependent for short-term memory?
Right, right.
And we just did it recently. We're like, okay, well, that's crazy. Didn't know. It seems silly, but it's kind of like there's a lot more to be learned, even with the stuff that you think that you already knew everything about.
Right. Yeah. Yeah. And the other thing that tell me if it was unexpected from your part, but it was interesting when you started looking at the tissues, and it was like, okay, well, you know, if we screw around with the neurons, then something happens. But then the hypodermis came up, and I guess first tell us what the hypodermis is.
Yeah, so the hypodermis, I think it's been the long ignored tissue in C. elegans. So people thought of it as just kind of like a structural tissue that secretes the cuticle, and part of that's a kind of weird name, hypodermis, which you don't even hear about that in other systems, right? And so, and for years we kind of I think we just totally ignored it. And it turns out part of the problem is that it's actually so important that if you mess around with it, it kills the animals. So a lot of people, for example, DAF16, when I was in Cynthia's lab as a postdoc with Milena Hansen, when we were making overexpression of DAF16. And we succeeded in doing that in lots of tissues, but not in the hypodermis. And we kind of didn't think about it at the time. And it turns out later we discovered there's another transcription factor called PQM1 that kind of is like the anti-FOXO.
Right.
And if you have too much DAF16, it keeps PQM1 out of the nucleus, and it can't do its job of allowing the animal to develop.
Right.
Okay.
Right.
And so, but it turns out, when we did transcriptional analysis of the hypodermis, that's when we discovered it's really like a metabolic tissue. People used to think the intestine was the only major metabolic tissue. It turns out the hypodermis might even be more important.
Right.
Or at least equal. And so that gets us to this question of, well, what is it really like? And that's what led us to ask the question, if you compare it transcriptionally to human tissues, what is it most like? And that's where we came up with liver and blood plasma, which was crazy.
Right.
Yeah.
Right. But then suddenly it begins to make sense.
Exactly.
It doesn't look like such a bizarre connection. So there's signaling. Is there signaling going from the hypodermis to the neurons?
That's right. And it's a brilliant tissue for that, because it's basically a bunch of, not one syncytium, but largely syncytial tissue. So basically, if you have a signal, you could actually get a signal really fast to the rest of the animal very quickly.
Right.
Which I no one's proven that. It's one of my favorite hypotheses about like why it's so important, because it's just basically it doesn't have to jump from cell to cell to cell. You could actually wash.
So is there a parallel then between? I mean, you have this kind of signaling between cells through neurons. We know that we have that as well.
Yeah.
And then if we signal, we have our circulatory system.
Exactly.
Yeah.
So in mammals, you might get the same effect as what we see with this signaling that goes from the hypodermis to neurons through, for example, liver and then to the blood plasma, which then might go to the brain.
Right.
Right? And so it's really parallel. In that case, it's more of a parallel, like an analogy rather than the exact homology, because we think it's different molecules. But you can imagine the same kind of, you need to keep your metabolism working, and you need to signal to your brain to keep looking for food. And the best way to do that would be start with the metabolic tissue and send the signal to the brain.
Right, right. Well, it's a fascinating story. Thanks very much for telling us all about it, and
My pleasure.
good luck following up with it.
Thank you very much.
Thank you.